Thank you so much, Claudia, and good afternoon to everybody. Let me thank the organizers for giving me the privilege to present this exciting meeting. What I can consider a sort of peculiar perspective, namely that of working in an academic setting in the field of CAR T cells, and that's my conflict of interest relevant for this presentation. So one question that you could immediately ask me is why to invest on CAR T cells when commercial products are going to be immediately available. And I try to offer you some explanation, namely because we wanted to have a drug product manufacturing process immediately available for our patients. The second reason was that of trying to investigate questions not addressed by sponsored trial. The third reason was that to try to address unmet medical needs, and in this regard I will present you some data on a solid neoplasia, namely neuroblastoma. And finally, we were challenged to prove whether the virtuous interaction between academic centers and companies so successfully in US is possible also in Europe, and in particular in Italy. So we started to work on two projects. One is that of CD19 targeting CAR T cells, and thanks to the collaboration that we have with the Belgian pharmaceutical, we decided to include in our second generation construct also safety switch, in particular the inducible caspase-9. For those of you who are not familiar with inducible caspase-9, this is an interesting safety switch. It's humanized, it's not immunogenic, and it is characterized by a very fast mechanism of action, as you can appreciate in this cartoon, and it can be activated by a dimerizing agent. And we decided to invest on a retroviral platform, also because we wanted to address in a certain way whether the viral platform or the costimulatory domain are more important for the effect and the function of CAR T cells. And we started a very demanding preclinical work for preparing the dossier to be approved by the Italian regulatory agency, clearly demonstrating a lot of in vitro question, in particular addressing the potency assay for not only the lytic effect of CAR T cells, but also the possibility that these CAR T cells can be killed by the activation of the suicide gene. We then move to the animal experiments, clearly demonstrating the efficacy of our CAR T cell in the animal model, but also getting what we believe to be an important answer to a relevant question, namely that if we re-expose the mice to a second infusion of the leukemia cell lines, the CAR T cells were able to clean and that also the second infusion of the leukemia cells. And in view of this experimental data, we designed a phase 1/2 clinical trial, and these are the relevant inclusion criteria. They are rather similar to those you have learned from the previous presentation, and they are also present in the company-driven trial. And this is our product manufacturing process. In particular, we are able to generate our CD19 CAR T cells at a median time of 28 days after the beginning of the cell culture. As I mentioned before, this is a phase 1 and 2 trial targeting 3 different dose levels in the phase 1 portion of the study, 500,000, 1.5 million, and 3 million of cells per kg of recipient body weight. Patients are prepared with a lymphodepleting therapy consisting of the combination of fludarabine and cyclophosphamide. We have already completed the phase 1 portion of the study, and we have also already treated 2 patients in the phase 2 portion. And you immediately realize from this slide that we treated a very advanced populations indeed in the setting of BCP-ALL. 8 out of the 10 patients had already failed previous stem cell transplantation. And in this last column of the cartoon, you can see the percentage of leukemia blast at start of the lymphodepleting therapy and not at start of the patient's enrollment into the trial. We have been able to generate the CAR T-cell product in all patients with a very good viability above our release criteria. And again, you can see that also the percentage of the transduced cells was very high, with the only exception of the patient number 3, in which the percentage of transduction was in the order of 21%. And by the way, I anticipate that this was one of the 2 patients who didn't respond to the therapy. The phase 1 portion of the study showed that the drug product is very well tolerated, and among the 10 patients with BCP-ALL, 8, so 80%, reach a complete remission with MRD negativity. And these data are more or less similar from the median percentage of CR and MRD negativity that you can extrapolate from the published studies so far. And when we look at the CAR T-cell expansion, we demonstrated a nice expansion both through the flow cytometry monitoring, but also through the qPCR monitoring, with a peak reached around 2 weeks after the drug product infusion. The waterfall plot of the patient is reported in this slide, and you can see that only 2 patients, namely patient number 3 and number 10, didn't respond to the infusion of the CAR T-cell product, and some of these patients have a very nice and sustained remission over time. So what about our future plans in the field of hematological malignancies? We want to invest on bispecific CAR T-cells, as other groups already mentioned during this wonderful afternoon. We are working to explore the efficacy of an alternative cell platform, namely CAR-NK cells. And we decided also to invest on the 2 most challenging scenarios, namely CAR for T-ALL and CAR cells also for AML. But as I mentioned before at the beginning of my talk, we also wanted to explore the efficacy of CAR T-cells in a solid tumor, namely focusing on neuroblastoma, which is the most frequent extracranial solid tumor of childhood. And you can see from this cartoon derived from a study published by COG some years ago that the prognosis of these patients in the metastatic phase is still unsatisfactory, at least for the pediatric standards. And when a child with neuroblastoma relapse, he is substantially without any chance to be rescued. So we prepare, in this case, a third-generation core construct, speculating that including two co-stimulatory domains, namely CD28 and 4-1BB, could improve the efficacy of the CAR T cells. The lab group demonstrated with a very extensive preclinical work that this was the best combination able to reach a sustained control in the animal model. And after having prepared all this data, we obtained the approval from the Italian regulatory agency for starting a phase 1 and 2 clinical trial with a rather original design, and in particular I'd like to acknowledge the contribution of Anne-Marie Mosley in helping us to identify this type of design, in the sense that it's an escalating, de-escalating strategy according to occurrence or not occurrence of toxicity. And we started at the dose level 3 with the possibility to increase to dose level 4 and 5, or to decrease to the dose level 2 and 1. And again, the patients were prepared with a lymphodepletion. We have completed the phase 1 portion of the study, and again, we enroll into this trial very advanced patients, stage 4 disease, having failed 3 to 4 lines of treatment with a very resistant disease at time of enrollment. In the phase 1 portion of the study, we didn't record any dose-limiting toxicity, so the final dose level to be employed in the phase 2 portion of the study, which will be also open to newly diagnosed patients in remission at the end of the conventional therapy, with the hope of improving the possibility of eradicating the disease. We didn't see any relevant toxicity, although some patients had either grade 2 or grade 3 CRS. Despite the fact that we work in a solid tumor, you can easily appreciate from this cartoon that through the flow cytometry monitoring, we were able to document the expansion of CAR T cells, accounting up to more than 50% of circulating T cells in peripheral blood. And we were particularly pleased to see that these cells are able to localize also in the bone marrow, because this can be instrumental for clearing the bone marrow metastasis of neuroblastoma. When we look at the cytokine production in these patients, we saw that there is a nice production of interferon gamma, IL-6 and IL-10, and the expansion of CAR T cells was more or less similar across the 3 dose levels, the dose level 3, 4, and 5 that I mentioned before. That slide shows the waterfall plot of each patient with measurable lesions, and you see that 2 patients reach a complete remission through the infusion of CAR T cells. And looking at the data on how this remission is sustained over time, you can see that there are some patients who are able to maintain a response, and notably when we infuse some patients who initially lost the response with a second round of CAR T cells, these CAR T cells were able to induce a new response. And just to finish, some images of the tumor mass. You can see in this cartoon before and 6 weeks after the infusion, the shrinkage of the tumor mass, and also the clearance of a bone metastasis in the skull. And for those of you who are not expert in the field of neuroblastoma cells selectively uptake a radionuclide, which is MIBG, and you see that the response can be also clearly determined at evaluating the uptake of MIBG. And in this particular case, where there were metastases before the infusion of CAR T cells at week 6 and 12 after the infusion, there was a clearance of the cells. So I'd just like to thank people working in my group, in particular Dr. Quintarelli and Dr. Del Buffalo, who were really instrumental for running these trials. Thank you.